So here we have broccoli, cantaloupe, corn, and some peanuts. How many of you know which one is the result of genetic modification? How many of you guys think it's the broccoli? Raise your hands, come on now. So you think it's the broccoli? How many of you guys think it's the cantaloupe? Corn? What about the salmon?
All of them.
Very nice. Okay. So you're correct. It just so happens that all of these are genetically modified. Okay. Last semester, I took human biology and human nutrition, and they both discussed genetically modified foods, and I learned a great deal about the subject from these classes. Today I'm going to share my knowledge about genetically modified foods with you. So hopefully you can take this and incorporate it into your life. Genetically modified food is a major product in today's food industry. Everyone eats it, so it's important to know what your food or how your food is created. From this presentation, I hope you take with you the knowledge of what classifies a genetically modified food, how that kind of food is made, and the possible benefits or dangers that exist through its use. The first point to cover is what is a genetically modified food. According to the textbook Nutrition Concepts and Controversies, genetic modification refers to intentional changes to genetic material of living things brought about through a range of methods, including rDNA technology, natural crossbreeding, and agricultural selective breeding. Not many recognize the term genetic modification as being natural, but it can be. Natural selection and cross-pollination are part of natural crossbreeding, which involves no human interaction. The second method, agricultural selective breeding, is the oldest way humans have interacted with crops and farm animals to produce an improved offspring they needed. This is how the broccoli and cantaloupe I showed came to be. The last method of genetic modification is rDNA technology, and is the most associated method. This method uses analyzing DNA to cross specific genes between two organisms. I have an illustration that shows that compares the difference genetically between selective breeding and rDNA technology. As you can see, selective breeding allows a desired gene, that is to be passed down to a new variety, but it also passes down genes that are not needed, as you can see in the yellow. With rDNA technology, scientists are able to focus on the desired gene that they want and implant it into their new variety without any other genes unwanted or needed in the new variety. So rDNA technology is often confused with being the sole method of genetic modification, because rDNA technology is also referred to as genetic engineering. So to keep them straight, and not to be so confused between the two very similar terms, just remember that genetic engineering and rDNA technology is just one of 3 different methods of genetic modification. So now that you know what genetic modification is, it is time to explain the possible benefits and dangers associated with this type of food. The reason genetic modification exists is because of the desire to improve food, but some wonder if the benefits are outweighed by the danger. Nature has been weeding out weaker varieties of plants and animal life for centuries, and humans took it to the next step with selective breeding. This allowed farmers to make offspring with improved characteristics, such as high growth rate, high food yield, and or disease resistance. An example of improved growth rate is the salmon shown on the first slide. Genetic modification or rDNA technology, or genetic engineering or rDNA technology, vastly furthered improving food in a variety of ways. An academic journal listed the first benefit from this technology as it was providing crops so that there's no pesticides or herbicides needed to be put on the plants. This made it safer for people to consume without affecting overall crop yield. Corn was an example of this. The same source informs us many genetic engineered food have enhanced nutritional traits, and many more can be developed to improve flavor, texture, and shelf life of fermented foods. I learned in nutrition class that rice has also been altered to include beta-carotene, which is a good source of vitamin A, so they can give it to developing countries with vitamin A deficiencies. The main cause for many genetic engineers was to solve world hunger. With population, Theodore Frederick, agricultural expert, said, production is not keeping pace with population. So with genetic engineered foods, production can be improved to change this problem. The possible dangers about using this new technology is that there's not enough information about it. Genetically modified food is considered marketable if testing shows no signs of it being unsafe. Researchers believe that the lack of evidence that genetically modified food is unsafe cannot be interpreted as proof that it is safe. This view is supported by Conrad G. Brooke, an bioethicist at the University of Ontario. When it comes to human and environmental safety, there should be clear evidence that the absence of risks. The mere absence of evidence is not enough. Furthermore, the danger of not knowing the safety of consuming our DNA products over a lifetime and being introduced to allergens from other species can be a risk. There are many other concerns about genetically modified foods, but they all revolve around the fear of the unknown. Today I have discussed what constitutes genetically modified food and given some information about its benefits and safety concerns. My hope is you take the info provided and use it to analyze food that you see in your daily life. So the next time you're at a grocery store, see how many genetically modified foods you can point out.
Frankie, what did you think?
I thought you were very clear on your topic. You knew what you were talking about the whole time. I thought you could have used a better attention grabber than using the audience. It doesn't work as good because not everyone participates in it. I liked how you actually told us where you got your stuff from. You didn't just state it. You said, like, you gave us quotes, you gave us an example, and you showed us how, like, you learned it and you had an interest in it. And other than that, I think you could have spoken a little bit louder. Other than that, you did good.
All right, yeah, I thought that the introduction, it's another one of those survey things. It's not necessarily a bad thing to do that, but you need to condense it because you're going through like 4 different ones on that. There's got to be a simpler way to ask, that kind of question and still not get a response from everybody, and then you could just get right to the thing. Actually, all of them have been genetically modified in some way, shape, or form and get to the point a little bit clearer. I thought you had a very good statement, but that was the only part of the opening that was problematic. Everything else was really sharp. You had a good, clear thesis statement. There's an excellent preview. You try and give us some justification about why we're talking about this particular subject from the very beginning. So that was pretty sharp. Hang on one moment.